Computer. All right.
All right, hi everyone. So welcome to another one of our TRALF talks, where we're really lucky today to have someone who I'll have to say actually really shaped my research career from a very early stage, Professor Olaf Sporns from Indiana University. So I had the pleasure of meeting Olaf for the first time back in, we just realized, 2014, which seems like 1,000 million years ago now, at a conference in Brisbane. But then I've had a number of other opportunities to chat with Olaf over the years. In fact, we actually got to collaborate on a project back in 2018 or so, which was really fun. It was kind of my first real foray into dynamical systems and trying to infer dynamical systems sort of insights from neuroimaging data. And it was just so much fun to work together. Um, Olaf, I'm sure I've told you this before, but your book Networks of the Brain just kind of opened my eyes to the kinds of answers that I wanted to get from a nervous system. And I think as a young fledgling field, we often forget that not all answers are created equal. And we often kind of get really sucked into the map that we have. And one of the
Mhm.
things that we've been talking about a lot recently in the group is what should our map even look like? What are the elements that we really want to see? What are the kinds of factors? What are the kinds of data? What are the kinds of questions we should be asking? And you really helped sort of accelerate that process for me, and so I'll be forever grateful for that catalyst. So anyway, that's my way of saying that I'm extremely happy to have you here today. So I'll remind everyone that the idea is that what we've asked Olaf to do is to join. He's going to give us a couple slides giving some context to the conversation, and then what we'd really like to do is learn about what's shaped Olaf's thinking, how he sees the questions that he goes after, how he drives his research career, and hopefully have a good chat together. So yeah, welcome Olaf. Great to have you.
Fantastic. Thanks so much for the introduction. And so as I was preparing for this sort of unusual meeting, right? I mean, usually we just, we have 40, 50 slides and we go through them in sequence and so forth. As I was preparing for that, I was thinking, where did my thinking really crystallize? Where did all those ideas about networks, complexity, dynamics, information, where did they come from? How old are they in my case? Um, and it got me to think back to my student days when I was an undergraduate, actually in biochemistry. Believe it or not, I started wanting to solve protein structures. That was my dream as a high school student, and then as an undergraduate. So I learned a lot of biochemistry in my undergraduate years, and then went on and joined more or less by chance a totally new endeavor, which was neural networks. This was sort of the mid 80s. We're talking about 40 years ago, I'm afraid to say. And that was really not a clever career choice because there was almost nobody working on that stuff. It was totally new. It was very much outside of my own comfort zone, but it was exciting. And for whatever reason, I stuck with it. So that led me on a path that actually ended me up. Let's see if I can share my screen with you real quick. Where we are today, which is in an arena that, of course, you know very well, is now full of conversation and work and analysis and experimentation on brain networks, really at all levels, from invertebrates to the mammalian nervous system, from humans to animals, from structure to function. And I use this slide in my talks typically at the very beginning just to set the stage and remind people how many different domains of research and neuroscience are now pervaded by networks, really. And I want to remind myself and you that this was not always the case. This has only been about 10, 15 years or so, maybe 20 if we really want to stretch it. But before that time, networks really didn't play a big role, at least not in the way that they do now. So in my own career, I invested pretty early in networks, a long time before the field really started. And it was really not a very safe career choice to make. Perhaps we can talk more about career choices later on. In any case, I ended up working as a postdoc with people. I was very fortunate to work with people who were really extraordinary intellectuals really, like Giulio Tononi and Carl Friston. We were both, the three of us were postdocs together in the early 90s. And so many of the ideas that sort of formed the basis of my own work came from collaborations with them, endless discussions, drawings on the blackboard and computer programming, and so forth. So a lot of the stuff came from, in my case, from the early 90s. In fact, during my PhD, um, a phenomenon was becoming very fashionable. That was 40 hertz oscillations in the brain. People thought that those oscillations were linked to the binding problem, how neural populations coordinated their activity, and perhaps even consciousness. So I set out to code up a model of the visual cortex. What else is an ambitious graduate student going to do but wire up some cortical columns, tens of thousands of neural elements and connections between them, and then inject random activity and see how the activity evolved in response to a stimulus. I'll show you this movie, which I made in using technology that is actually extremely arcane, like 16 millimeter movies and VHS tapes, but it still plays. I'll show you a stimulus. This one here, vertical elements in a visual array. As soon as I move some of these coherently, you are seeing the outline of a square or a diamond. We call that phenomenon perceptual grouping. If we show the stimulus to our visual cortical model, we're seeing neural activity percolating at first at random and then gradually ramping up and forming a coherent synchronous assembly of neural population activity that was orchestrated by connections that linked these neural columns together. When I carried out this work in the late 80s, in this case, over 40 years ago, what struck me was two things. For computational neuroscience models, you really needed just two ingredients: the dynamic equations, which governed how the neuron or the neural element behaved over time, and a coupling matrix, which was essentially coded up as a graph, as a matrix of connections. So even back then, in the early 90s, the idea that connectivity was equal to a graph started to get hold of me. A little later, we analyzed a matrix of connections in the macaque visual cortex, the only data set we had available to us at the time. And we pursued an angle that was looking for clusters in this matrix. We found a couple that you see here. To identify these clusters, we used a cost function that was based on functional connectivity, essentially functional connectivity, the covariance matrix of the system's spontaneous dynamics. And it occurred to us that this matrix looked clustered. In other words, there were two big clusters evident in it. We now call these modules, and modules have become very fashionable in network neuroscience. This work dates back to the year 2000. Simultaneously working with Giulio especially, we developed some tools for multivariate information theory that allowed us to characterize how information was distributed in a larger system. In the process, we developed something called integration, which is now called total correlation, which quantifies the total amount of shared information in a given subset of elements, and leverage that to define a measure of complexity, where complexity is defined in our framework as the interplay or the balance between integration and segregation. A system is complex if at the same time it carries information that is specialized, and that information can also become integrated. That is in a nutshell what the formalism implies, and that is the idea behind our particular formulation of complexity, which we developed in the last century. Last idea here is, is there a connection between complexity and certain types of connectivity? To address that, I built an evolutionary algorithm back then, essentially taking a random network, rewiring it at random just a little bit, evaluating the offspring for how much complexity it could generate, keeping the winner with the highest complexity, getting rid of all the others, and again rewiring, starting the process again. So variation and selection basically ongoing in a computational algorithm. And when we do that, we find that driving complexity higher and higher ends us up with a system that has clustered architecture. You see it at the top at the connection matrix. That's what we're rewiring. At the bottom is the functional connectivity, if you wish, the covariance matrix of the system, which has sort of like a clustery aspect to it. So complexity and modularity seem to be interestingly linked. At least that came out of this computational exploration. And all of this work kind of gave rise to a framework that I'm still very fond of, but it was basically in place, in my case, at least 25 years ago, that complex brain dynamics consisted of episodes of coherent or integrated activity, and then incoherent or decoherent segregated activity, which was carried out in variable subsets of neurons and brain regions. We could quantify all that interplay of coherence and decoherence with information theory, and systems that contain specialized elements that could also integrate. So that's the synergy of specialization and integration. Those systems were highly complex. And if we have a modular system, we get complexity for free. So that was kind of where things stood for me coming out of my very long 10-year postdoc. And where my thinking stood at the time, working very closely again with Tononi and Friston. And all those papers down below that I'm still very fond of, none of them were highly cited when they first came out. They kind of, they were sleepers. A handful of people got interested in the work, but not a whole lot of people were excited about it. So for a long time, we kind of worked in relative obscurity and developed these sorts of ideas. And it turns out that for me, at least, the ideas remained attractive enough to pursue them for the next couple of decades. And now we have a lot of data, right? Things have changed. Back then, we had almost no data. So theory was the only thing we could do, simulation to some extent. But we had no real, there was fMRI, but there was no functional connectivity yet. There was no real notion of brain dynamics, at least not in the mainstream of neuroscience. Anyway, where are we now? This is the last slide. This is sort of my summary slide, typically. The ingredients that I feel are important if you want to understand the brain. There are probably several others, but the ones that have played a role in my own work are in this little summary. On the one side, networks, of course. Connectomics, right? The connectome is actually turning 20 years next month. Kind of entered the conversation in 2005. And there's a lot we can do with graph theory and with finding modules and their interactions, the element of cost and efficiency. On the other side, dynamics on top of these networks, the notion of flows and manifolds, very important. Synchrony, coherence, and dynamics, not only of neural activity, but also of connections. Connections themselves change over time, and we often forget that communication patterns unfolding across a network are also dynamic. The interplay between the two gives rise to lots of interesting phenomena. We can investigate dynamics on networks, but also dynamics off networks, how networks change over time, and how that change in turn gives rise to changes in dynamic architecture. And then on top of that, information. That's probably the closest I've come in my own work to the old idea of computational information processing in the brain. In some ways, the interplay of networks and dynamics is carried out in the service of creating information, of creating rich and always time varying, coherent and not coherent patterns that are somewhat redundant, somewhat synergistic. And much to my great joy, really, the last few years, there's been a resurgence in multivariate information theory as applied to the brain. For a long time, there was not much going on, I would say, for about 20 years or so. It was fairly dormant. But recently it's been revived, and there have been a lot of exciting studies carried out recently on redundancy, synergy, and higher order interactions among neural elements. So that's nice to see. So that's kind of the magic triangle for me, and most of my work has been sort of on one of these vertices of the triangle or multiple of them. And I feel that, those three ingredients, if we could understand them better, and we're still at the very beginning, I think, in our field, would buy us a lot of insight and understanding into how the brain is actually working. So that was all of my introduction. I'm going to stop sharing so that we can all see each other a little bit more easily. Um, so as you can see, I've taken a fairly long journey already, and not all of it was very linear. Um,
Huh.
a few strange curves and turns in there. But it's gotten me at least to this point, and so I'm wondering,
So let me jump in there. So that was really
yeah,
great, Olaf, and thanks for the kind of bird's eye view of kind of where you've come from and where you're going. I might take you all the way back. I'd love to know what it was like to be in the room on that whiteboard or blackboard, maybe it was back in the day, um, with Carl and Julia and others, and kind of hear about what it was like back when you were sort of carving out these questions. I think this is something that is a kind of real experience for a lot of us as scientists, right? We can see something that we think is an interesting question, but we're not quite sure how to put our finger on it. We're not quite sure
yeah.
the right question to ask or how do we communicate this to someone else? And so I'd love to know, what was that like? Would you come with an idea like a musical band? Someone comes in with a riff and then you sort of play
Yeah,
around with it, or like what was it like? Tell me.
I've never been in a band, but I suspect it's a little bit like that. And
Mm,
really, a lot was, we had no phones back then. We had no internet actually in the very early days. No distractions. We really did have just chalk and not even markers, as I recall, and paper and pencil and lots of coffee and cigarettes in the
yeah.
case of Carl. Um, so and we went back to the same ideas over and over again. Is what I also recall. We it was not a matter of we had an idea and there it was and we were done with it. No, we came back to it.
Mm,
Um,
mm,
the paper on complexity, which ultimately came out 31 years ago now. I think Julia and I must have spent hundreds of hours uh, going back and forth over various formulations, testing them out on ridiculously small network models and with really, very simple Fortran and then later MATLAB code, trying to figure out what the ramifications of these measures were and how they would play out. We had no way of scaling up things. We had very little computation back then either. So it was more like that. And Carl had a wide-ranging intellect and was just beginning to code up SPM. I remember the very first version back then, and she introduced me to MATLAB actually, I think. And so it was sort of like the other thing was there was not a lot of external pressure on us to produce. So there was not the
mm,
hunt for the high impact paper. There was not that pressure to publish in Nature or Science. In fact, I've never had a Nature or Science paper in my career. So, we could develop these ideas fairly much at our own pace. And we had the luxury of having a little bit of time to let them mature and to actually let them ripen, if you wish,
mm.
and really be sure about them before we actually put pen to paper.
Yeah,
So
and so a lot of that then would come down to kind of your internal level of satisfaction with
yeah,
the answer you had, which I think is something, we kind of often, like as you sort of say, we sort of externalize this a little bit. We say,
yeah,
if this passes muster and gets into a high journal, it is therefore a fact
yeah,
worthy of
yeah,
kind of consideration. Whereas if we don't actually think ahead of time or like take seriously whether or not our colleagues or ourselves are comfortable with the level of the answer, then we're kind of maybe we're not building the foundation in a way that's stable enough to actually reach the kinds of questions you got to. Um, yeah, Eli, what did you want to ask?
I think Julia's question was first, but sure, I'll go. How you going, Olaf? My question, I guess, is I'm kind of want to tap into your sort of experience that spans such a long time. Like, looking back on where you were in sort of like the late 80s, early 90s, and your decision-making processes there, like which sort of avenues to go down. You talked about sort of eventually information and complexity and networks. How do you view that now? Do you feel like you really sort of you know, found some really good avenues that obviously turned out to be wonderful for the community. But are there others that you felt like, oh, if only I'd sort of pushed a little bit in that direction? that would have been cool back then, and now I've sort of found my way back around to it now. Do you feel like that? I don't want to say it's like mistakes, and like I was curious about your decision making process, like now relative to how you
I
had it
didn't
then.
have a, I don't ever remember having a decision-making process, to be honest. I was ridiculously unaware of my future. I just kind of did things that I liked doing. And again, I had sort of the luxury of being able to do that because there was not a lot of external pressure on me personally. You got to do this or career path or whatever. I was just doing things that I felt, um, again, fit with my intellectual mission that I had, which was to figure out something about how the brain worked. I actually did a lot of site tours, if you wish. I had a wet lab for a while. I did video microscopy of neurons, published a few papers, even had a couple of postdocs um, in trying to find empirical evidence for this notion that connections were changing dynamically on a fast time scale. And so I spent many, many, many hours looking at real neurons growing in a little petri dish and taking videos of them and things like that. I also had at the same time a robotics lab where I built robots with some of my colleagues to study things things to do with embodied cognition, the notion that which was a very fringe idea back then, the idea that cognition requires interaction of the body and sensory organs and effectors with the environment, ongoing behavior as a source of information, as a source of interaction. Um, and I pursued that with robots. In fact, the reason I got hired here in 2000 for my first real job was the robot work. That was my main bread and butter work at the time. Connectivity was still extremely small. There were like 5 people. It was, Klaus Hilgetag and Wolf Kötter and Macintosh, myself, um, and Carl, and that was it. The first brain connectivity workshop started, I think, in 2002 or 2003. I was still doing robots back then for a living. I tried to get a grant from NIH, and thankfully, I am so grateful they turned me down. I tried really hard to get this big grant for doing the robot work. If they hadn't turned me down, I would have probably still be doing robots. Um, but they turned me down, so that kind of cut that off, and gave me the opportunity really to work on networks full time. This is sort of the mid 2000s, and so even a lack of success in the end turned into something positive for me at least.
Are these the Darwin robots, Olaf?
Yeah, this is sort of Darwin. These automata, we called them back then. We didn't actually like the word robot so much because my boss just didn't like certain words. Anyway,
Yeah.
automata, that I did that in my thesis work a little bit, and then later on we built real robots with wheels and cameras and
Were they
uh,
were they little connectionist robots? Were they cybernetic? Like, what was there must
well,
have
they
been
had they had
intention. Yeah.
they had sort of neural network brains that were usually run off base because we couldn't put enough computing power onto the platform.
Mm.
It was sitting on a desktop, and we were remotely. It was very slow, so we had to take an image to the camera, send it to the computer. The brain worked on it for a while, sent out a motion command. It took like several seconds for a single lurch of the poor machine. So ridiculous stuff, really, very rudimentary at the time, but it sharpened my perspective because it opened my mind to the notion that not everything happens between the ears, right? brain is not just the brain is not just in there doing its own thing like a computer, but it's really we're always coupled to the environment. I
Oh
am
yeah.
talking to a screen right now, so my neurons are very busy processing all sorts of sensory inputs, which in turn modifies my functional connectivity that I'm currently expressing. Right? I mean, that's resting state is a very weird scenario. It's the gift that keeps on giving. It's been very good for our field in many ways, but it kind of pushes us into a realm where we are really not interacting with the environment anymore. And in natural behavior, we always do that. And the robots really sharpened my sense. The robot work sharpened my understanding of that. Even though I've stopped it now and haven't touched the robots in 20 years, I still feel like that has taught me a lot.
Yeah, that lesson stays with you, doesn't it?
Yeah,
We
yeah,
we had Randall Beer on for a sort of more
yeah.
standard scientific talk last year, and his stuff, which I gather would have been around about when you were there
Yeah,
as
yeah.
well,
I
was he had this really lovely demonstration of one of his little automata now in a completely virtual sense that he trains them over with a genetic algorithm to be able to perform some function that he
yep,
sets up, and then he calculates the mutual information from elements of their structure to the
yep,
challenge that they're trying to solve. And he had this really, really great example where I think there was like this little motor and it could catch a thing that was smaller than its
yep.
mouth, and if it was bigger, it would move out of the way. And in one of the scenarios, he went through looking at the mutual information, and he couldn't find any mutual information between the activity of the robot's brain and the performance on the trial, even though it was a completely successful trial.
It was between the sensor and the actuator. There's no
Yeah, thanks for
information being
stealing my
shared.
punchline, Eli. Bloody hell,
No, that was what he was showing in the sensor.
step it on my lines. But you're right. That's the idea, right? Is that this little robot in this trial was sitting out of the way of the pellet that would have crushed it. And so the mutual information was in the X location of the animal's body. It had nothing to do with its brain. It didn't need to
Yeah,
brain that
yeah,
trial. And I
yeah.
think this is such a lovely, simple example of the fact that, as you've just said, we kind of go through as neuroscientists, putting the brain up on a pedestal and saying, where could we find the brain's solution to this problem? When the brain is actually a helpful sort of assistant in solving really embodied problems that we have in our lives. And yet we don't think of it that way. We often put it as kind of like this mind that's out there kind of
Yeah.
on its own solving these
And I think
symbolic
it
problems.
goes,
So yeah.
and I think it goes together with sort of taking an evolutionary perspective. I don't know much about evolution. I don't want to, embarrass myself by talking about evolution here, but I try to read about it, and it's also a never-ending source of inspiration for me.
Hmm.
Sort of asking where does it all come from? Why do we have brains in the first place? Why do we have sense organs and effectors? And how did that all arose, and why are we having bilateral symmetry? Why are we having all these sort of architectural features that we've carried along for hundreds of millions of years? And it's very simple things that play a big role, and predator-prey relationships, remote sensing, the ability of carrying out complex motor routines, being embedded in an environment, being subject to gravity, being subject to external forces that want to throw you on the ground, and you have to resist that. I mean, all these things that nervous systems have to deal with are important, and we forget them when we think of the brain as a computer, right? And that's to me, it's like I've never liked that metaphor. I've never liked that way of thinking of the brain, and it still riles me that it's so ingrained in our community.
Yeah, I've just been doing a bit of a deep dive back into Michael Turvey's work. He passed away
Yeah,
last year, but he
yeah.
was, I think, a beautiful proponent of this.
The ecological psychology. Mhm.
Let's look for kind of ecological explanations for something that seems otherworldly, and otherwise would require like multiple degrees in physics to solve as a mathematical problem. Yet it could be solved now by this simple coupling between the rate of change of something on your retina and the rate of change of your body in the world. And if you can make those things match, all of a sudden you've done the equivalent of this incredibly complicated partial differential set of partial differential equations. So I'm really attracted to this kind of looking for simple solutions. That's what, evolution's good at, right? It doesn't necessarily find optimal solutions all the time, but it
Yep.
finds good enough solutions that solve the problem for you. It gives you the tricks. I really,
And
I,
it's
I really love
often
it.
and it's often impossible to go back and start over, because what you have is what you have, and you got to build on it. And even if it's not optimal, it as long as it's good for survival, it keeps on going. So the way that this line of thought has informed my own work more recently is to do with the sort of the aspect of economy and brain architecture. Like, why do brains look the way they do? Why is our brain wired up the way it is? Why are the connection pathways laid out in the specific pattern?
Mm,
Why do circuits have the kind of geometry that they actually have? And I'm very intrigued, have been for a long time now, by this line of work that sort of says um, we brains have had to obey very very serious constraints, especially on cost. By that I mean volume, wiring length, yeah, energetic cost, actually being able to power the architecture and maintain it. And those constraints they're inescapable. Evolution, an organism cannot work outside of those constraints. If the brain does not geometrically fit in the room that it has, then it can't be built. So these sort of geometric and cost constraints have probably played an enormously important role in shaping the brain that we currently have in terms of how it's built and how it's operating. Which means, in other ways, that there are probably brains that are possible, in principle at least, in a computer to build that are much, much more powerful, but you just can't build them in the real world with actual lipids and proteins and ATP and glucose and the volume that you have available to you, the resources that you have. So our brains may turn out to be extremely suboptimal in the end, because of all these constraints that are put on them. You couldn't actually break out much from that, and so the kinds of brains that AI systems are now giving rise to are very, very different in many ways, because they're not subject to those cost constraints. They're not. There's no such obstacle in terms of wiring length or energy consumption for that matter. It can just be whatever it wants to be. That's not possible for biological organisms.
There was a really lovely paper just came out recently from Anil Seth, who it's a brain behavioral sciences paper. It's one of these kind of gigantic kind of 50-page things where he gets to kind of lay out his logic of a problem and then invites people to respond to it. But he's arguing essentially along these lines of what is AI right now, as we've currently kind of termed it, neural networks, let's say, writ large, versus what is biological intelligence, and how do we kind of think of the difference between them? How will we ever understand whether or not an AI is conscious in the way that we're conscious? And there's a lot of terminology that has to be built out here. But at the core of it, what he's arguing for is something along the lines of what you're just talking about. We know that biological organisms can process information in the way that we can, but we don't quite know how. And we know that AIs can do certain tricks and tips, and we really do kind of know at least what we've done to build them. We kind of know the architectural constraints. But there's this question at the core of it is, what if there's something in the biology that we don't even know about yet, that we haven't discovered or that we haven't considered as important for that process? And he gives this lovely example of nitric oxide. And nitric oxide is this chemical that we often forget about, but it's deeply important for cells. It's a marker.
I actually worked on that. Was my wet lab project, nitric oxide and plasticity. It's a retrograde signal, and it's a gas, so it goes through everything. There's no barrier.
Through everything, and there's sensors all over living cells. So it's not like just in the axon or just in the dendrite, right? It's all over the place. And so now, if you wanted to build that into your computation of a neural network, you'd have to build it again from the start. You'd have to say, now there's elements that can sense lots of different signals coming from everywhere, and they can have in this particular cost function and this, maybe a wave approximation or something of the diffusion that then has to be built into the message passing story that you've told yourself is the kind of core of the intelligence that's actually on display here. But we really at the moment don't know whether or not this idea of being able to respond to a gas is critical or not for actually being able to build intelligent life. It might not be in the end. It might be that computationalism is true, that you could have a invariant substrate that runs the process of intelligence. That's also possible, but at the moment we don't have enough information to know whether or not that's actually true. And it's sort of, I love these kinds of examples because they kind of bring you right back to the kind of naivety of where we are. We don't really know how it is that life works. And there's still so many questions to answer that are so basic to build into this bigger picture. I think having some confidence intervals around whether or not AI is conscious, it's sort of nice to kind of know where the edges are.
Julia, did you want to ask your question? We've kind of been off the track there from that for quite a while.
Yeah. Yeah. Hi. I have a question for you. I didn't know about your more wet lab background. It's really fascinating because my question is, how did you then go from more of a causal mechanistic understanding where you had, you could perturb the system and really come up with theories about how potentially these networks work in the brain to then move to data. I'm sure you were excited, as you said, to finally be able to test your ideas,
Yeah.
but then especially a lot of the papers I read from you are in fMRI, where we know there's, eons away from the real data because they're
Yeah.
inferential and indirect.
Yeah, I mean, that's a good question. I think in terms of my own sort of scientific biography, I've never let go of that mechanistic sense of that constructivistic sense of building something. Okay, I mean, I wanted to, again, my ambition as a very young, high school and then undergraduate student was to understand proteins and sort of, large molecules. And for that, you deal with models. The models quite often have to do with, like, these molecular models that you build in 3 dimensions, and they were fascinating to me because proteins were little machines. And if you made a change in a protein somewhere, it changed the way it worked, and sometimes in a very bad way. So that notion of structure coming first and function following it was with me, like, from my very early years as a student. I took it forward, I think, into the domain of computational neuroscience because when I started building my first sort of big simulations of neural circuits and then injected dynamics to see how they worked, um, what fascinated me was that I could modify the connectivity. I could modify the topology of the connections. I could just change the matrix that I loaded into the simulation, and I got very different behavior out of that. That was step number 2. Step number 3 was what you mentioned, the wet lab work, manipulating cells under the microscope using fluorescent signals and then measuring responses. Fourth step for me was the robots, because again, it was constructive. I was just over in the lab. I still have some old robots sitting around, and I reminded myself that I actually built these things in the basement of this building when I first started out here as an assistant professor. And again, it was building, right? You build a body, you build in combination with that an artificial neural network brain. It was a constructive exercise. And then you got something that you could study, behavior, movement, navigation in an environment, approach, avoidance of objects, things like that. And all of these activities um, gave me a sense of, causal mechanism and experimentation, actually, but using the computer quite often as my tool, right? So using using the computer as a way of instantiating hypotheses, if you wish, and then testing where they lead me. Um, so the idea of the connectome came from that because the connectome was lacking. We didn't have any data back then. I mentioned that. And the most important data for me at the time, and I think still today, is that basic architecture, the structure, right? How is it built? How is it wired up? And what are the building blocks? How are they connected with each other? And that was the idea behind the connector. We needed that, and now we have it. At least it's an evolving research enterprise, obviously, but we have made a lot of progress the last 20 years, getting a lot of these maps built, and now we have a foundation, I think, on which we can stand. But again, for me, the connectome is a causal, it's a mechanism almost. It's like it is physical things that we can verify are there or not there in the real world, just like stars or galaxies or rocks or whatever. They are physical things. We can verify they're there, and they have certain properties. And the way they are built and the way they interact is the foundation for everything else that follows, whether it's functional connectivity, whether it is ongoing behavior in the environment. Structure to me is fundamental. And a lot of people, I think, even very good friends in our field, I think they have everybody is entitled to their own views. I have mine, but I feel like that's still underappreciated. People sometimes treat constructs like functional connectivity as if they're real things, but they're not. It's just a covariance map, covariance matrix in most cases. It is the outcome of something structural. It is along down the road from a causal process. It is the end result of something that we are not observing directly. And I think we need to be aware of that at least. We can't always work on all levels. We all have to specialize and do certain things, but I think we need to be aware of that. And my whatever path I've taken in my career has always been reinforcing that notion that structure is fundamental. Anyway, that was a long answer. Sorry about that.
No, it's a great answer. It's lovely to
thank
hear the
you.
kind of genesis of. So I'm actually curious. I went back and reread your paper with Rolf and Julio on the kind of human connectome, the kind of call to arms from 20 years ago
Yeah.
on the sort of 20th anniversary. And I think it was actually really fun to reread, especially having kind of sort of dropped into the field halfway through that, where one of my kind of criticisms, I guess, or like reflections on the field was that it felt like we were taking the thing that we could map, the diffusion connectome of the cerebral cortex, as the network, and forgetting about all the other complexity that was really hard to map. And you've done some really beautiful work with Larry Swanson, mapping the kind of, connections of the hypothalamus to the rest of the system. And we could look at the thalamus and we could look at the cerebellum and all this other stuff. But when I went back and read your paper, you were actually advocating for that back in the day. You were saying we need a map of this complex web of connections, but it just so happens that the field couldn't do that yet. It was really hard. And as you say, technology's advanced, and now we can do this again and again and again. But I wanted to kind of hear your thoughts on whether you think as a field, the field kind of pushing into the thing that it could do as opposed to the thing the kind of complexity of the whole thing. Maybe there's like a subset that they tackled rather than the whole, which was really hard. Was there like challenges with that? Was it frustrating to watch this subset the part taken as the whole, or did you think, oh, this is a necessary step towards the bigger picture?
So there's multiple things in there. Let me see if I can unpack that. So first of all, getting the connectome out there and even published was actually a pretty hard scrabble. We had multiple rejections. I won't say what wonderful journals rejected the paper initially for lack of general interest, I might add, and but then persisted somehow to get it in, then we tried to get money for it. Actually, we tried to do some work in this area. Wolf and I went to Washington and lobbied for it. We couldn't get anything. For several years, there was nothing happening. And then, 2009, I think it was, the Human Connectome Project suddenly, like a thunderclap, appeared from the sky. I remember the day was like May 10th or something of that year when the call for proposals went out from NIH, and that was the floodgates opened at that point. Although I will say the term connectome has been used in so many different ways now that it's a little bit speaking as the guy who kind of defined it originally. I feel like the aspect of that is a structural map that has been a little bit forgotten along the way. So a resting state functional connectivity matrix is often called a connectome. That's not what it is. I mean, it's at least not by my definition of it. It is a connection map maybe, but it's not an ohm in the sense that ohms have to have they have to be verifiable, they have to be universal, they have to be complete, they have to be true for all time. It can't be something that you do every 5 years have a new ohm. The genome, ATCs and Gs, 3.5 billion of them in a sequence. Once you have it sequenced, that's it. You are not going back again in 100 years and you redefine it. The ohm is the ohm, and that's what it is. So the same with what the structure buys you is it buys you that it's physical, it's verifiable, it's real. You may not be able to measure it very easily, admittedly. That's a huge obstacle still. 3-dimensional, microscopic, macroscopic, it's tough. But the structural part of it is essential because that is something that remains invariant and should really be something that we actually converge on with multiple measurement techniques coming from different directions. Anyway, so that was good. That took off after a few years, and then now we have a lot of data, right? I mean, we have last year or so we got the adult, or at least one version of the adult Drosophila connectome, which is over 100,000 neurons and many millions of connections that have been mapped in exquisite detail. People are already working on that. Papers have come out or coming out continuously using that data sets. It's fantastic to see. I must say, I would never have imagined seeing that type of data set appear in our field so soon. It looked really daunting 20 years ago. To do anything of that magnitude was impossible to imagine, but now we have it. So I think what we are, if I can make one last point, one thing that's really been changing over my career is we had very little data originally, or sparse data, small data only. And that gave rise to really a lot of maybe theorizing and thinking about concepts more deeply and more slowly and more comprehensively. Now we have a lot of data, and in fact, we're inundated with data. And I worry sometimes that our field is too much invested in things like statistics and data analysis as opposed to understanding more fundamental concepts that actually that are inherent to the system that we study, not so much the data. You know what I mean? MRI data is great. I love it, but it's not really, it's one particular aspect of the brain that we're measuring. It's not the brain itself. And I feel like we're so busy now with such rich and voluminous and challenging data sets that perhaps we're forgetting a little bit that we need to at some point come up with Newton's laws, for the brain. No one has done that so far. I think Carl has come closest, but where are those big regularities that we can write down on the back of an envelope that are actually true? And I don't know whether we are closer to it now than we were 20 years ago. I hope we are, but the data is not always the answer.
Yeah, I think that's spot on. So that seems like the answer to my long-winded and convoluted question was, yes, it's good, but let's not lose track of the actual kind of map and take it for the territory. Let's actually kind of be thoughtful about what the answers ought to look like, and they're going to come back to an invariance to the particular measurement modality. It's not going to be that cognition is a bloody resting state network that's interacting with another one. That's just what the shadow of it looks like when it's played onto the corner of the cave. Let's think carefully about how that works. I think that's nice to hear. Julia, you have a question.
Yeah, I wanted to jump on the thought that you just had, Olaf, on the fact that we're moving towards more stats and data analysis and actually foundational understanding of what's happening in the brain. And I think, I don't know if you would agree, but probably one of the reasons why, especially people in MRI, decouple structure and function is because they observe, right, using inferential stats, say cortical thickness, and then they relate that like a structural network to a functional network, that sometimes these two things don't overlap. And so then they use this as an excuse, quote unquote, to say, well, I shouldn't use a structural backbone to understand functional connectivity because it's not always the map that will get me to the function. And I've seen that multiple times, even in diseases, right? They're like, well, in AD, for instance, function goes away before structure and sometimes, so why should I care about structure? So I wonder, yeah, what do you think about that? And I guess that feeds back to the idea that we need to think that this is step 7 of a chain, right? MRI is, as Max said, just the shadow of what's happening underneath. And maybe we should try and remove ourselves from the data and think more about models. And I guess that's what these labs here are doing in yourself as well.
Yeah. I mean, you're making a good point, but just that, structure and function can often decouple in empirical investigations, right? They don't necessarily move in an obvious way in sort of a parallel fashion. And then, both structure and function are actually complex constructs, right? I mean, what when we talk about, MRI, for instance, what does structure mean? Well, people do tractography and reconstruct pathways and so forth. But there's many things, as we all know, that it's a technique that has its own issues that have been written about and written about again. But there's also things that we're probably really lacking that we're just not measuring, like conduction velocities, which are in some ways structural parameters, like how what does the delay structure look like in the brain? We have very little information on that, empirical information, and things like integrity of pathways, physiological efficacy is something we cannot measure with MRI. By biophysical parameters that again are aspects of the structure that have to do with conductances and with efficacies, with transmitters and so forth. So there's things that we just don't have real good access to. So our structural models are very impoverished in many ways because they just rely on fiber densities or streamline densities and things like that. That doesn't invalidate structure though. It just means that we have an incomplete picture at this point of what the structure looks like, and the things that in a clinical condition that might change structurally, we simply may not be measuring them well enough to see those changes unfold, which then manifest in functional connectivity differences. Functional connectivity itself is often measured with fMRI and resting state or task fMRI, but the fMRI signal, I mean, I don't want to belabor it. It is what it is. I mean, it's great to have it. I'm so glad we have some way of looking at the whole brain non-invasively, but it is also it's impoverished, right? I mean, it's temporally and spatially blurred. We're not seeing a lot of the fine temporal detail that we know must be there from other studies. So we are getting a very partial view of the functional connectivity also. And again, I think these arguments are not, it's more about the insufficiency of our techniques and the fact that we are still working with data that's partly incomplete and partly just simply not measuring the things we should be measuring. That's why I find simulation and computer work so informative, because there you know what you put in. There are no unknowns in your code. There shouldn't be, or in the variables or parameters that you've defined, right? It is what it is. That's what you've put in. Then you can observe what's coming out, whether it's dynamics, whether it's behavior, whether it's a behaving robot, what have you, okay? Now you have the complete picture. It may not be an actual real organism. It may only resemble it superficially, but it's a constructive exercise that lays all the cards on the table. And it still can be very surprising, I mean, I'm starting to teach my class again next week, and I always use the good old, Kerr motor oscillator in my class as an example of here's a system that is so boneheadedly simple. It's one equation with one free parameter basically, and man, you can work that system and make it do things that you just would not believe. Okay, and that's the kind of thing that I think is very, at least educationally, very important to get across. So I think, it's a work in progress. We have to admit that what we measure today is not going to be what we're going to be measuring in 10 or 20 or 50 years. I hope not. If it is, then we're stuck. But, and again, I look back on my PhD education, there was no fMRI during my PhD, not at all. I mean, it didn't exist, came out of the woodworks in 92. And so nothing I learned in my PhD prepared me for that.
Myri, you have a question?
Yeah, related to that, throughout this talk today, you've touched on like some challenges you've faced in your early days and how things like data, which is something that you needed, has almost turned into something that is a challenge today. So I'm curious to hear what you might anticipate could be challenging in a technical sense in network neuroscience, but also in a broader general sense. Like you mentioned how back in the day you had no distractions and you were able to just think these concepts through. So I'm curious to hear, yeah, what you anticipate could be challenging in the future and how we could reconcile that.
That's a great question, and it's one where I could I don't at some level I don't know what to say, and at some level I also do know what to say, but I'm not sure I should say it. I mean, there's so many. I think the whole scientific landscape and the whole sort of what it means to be a scientist has changed a lot over my lifetime, certainly, and qualities and skills that are now very important were not important when I was around as a student or a postdoc. And I don't know if I don't know what to make of that. Or maybe that's good, maybe that's bad. I don't know. One thing, so some career advice I always give to my students when they get ready to leave, and a couple of them just finished up their PhDs this spring and are now postdocs. And so some of the parting advice I give them is keep an open mind, especially try to collaborate and work with people. If not collaborate, at least work with people, talk with people, be around people that are not like you. And I mean that in a scientific sense, okay? That are not all of the same type of, that are not all doing the same type of work that you're doing, okay? If all the people you ever meet are doing the same work that you do, you will never learn anything new. One of the great things in my career has been that I've always been around people that were very different from me in terms of what their skill set was and their philosophy was, and ideas sometimes to the point that I violently disagreed with them. But it was very important for me to always seek their company and also work with them. I sometimes jokingly say that in your social network, what you should try to do is maximize your betweenness centrality. Okay, by that I mean you should be the node in your network that connects different communities to each other. You have lots of connections, let's say, with MRI research and brain, whatever, but you also know the other person who works on, is an ornithologist, or who works on extinct mammals, or who works in astrophysics, or who knows what. And you will be surprised at the connections that come that suddenly appear out of nowhere that make you special, because now you have put together two different things. You have performed a synergy, right? You have taken specialized information from different sources and you've put it together. That to me makes not only for an interesting career, but also potentially for a career that's sort of a little bit apart from other people's careers, where you go a different way. And that can be a little risky sometimes, but on the other hand, can also be very rewarding. So,
I fully agree with that, Olaf.
yeah.
There's a really good bit of advice I heard when I was a young postdoc, which said you should make sure you're not going to the same conferences as all the people on the same floor as you, which I think is similar advice. Like, take your individual interest and differences and just like put chips on it and try to grow that thing so that it becomes more unique. Because the questions you get then are just fascinating.
Yeah.
So I wanted to pick your brain a little bit, Olaf. We talk a lot about evolution in the lab too, but we're not evolutionary biologists. We kind of, I think, in a similar camp to you. We like to be kind of evolutionarily constrained, kind of computational theoretical neuroscientists. And one of the curious things about evolution is that even though structure is the kind of building block of the way that the brain is formed, or a leg is formed, or a heart is formed, it's its actual functional outcome that matters at the end of the day. And so there could be multiple different ways to realize the same dynamics. And I think this is another lesson that computational modeling teaches us. Randy Beer's stuff is a lovely example from before. And so, you and I have both shared an interest in dynamics. I'd like to kind of push maybe the conversation a little bit towards how you're thinking about dynamics at the moment. Obviously fMRI, as you've mentioned, is a little sluggish. EEG can be a little bit noisy. Meg, gives you some benefits. Spiking data looks completely different. Do you have thoughts on which is the right resolution to look at, or do you try to come at it from this real zoomed out multi-scale perspective where you think there's different insights from each one, even though the dynamics look really different in each case? So I don't have any really great poignent questions. I just like to know your thoughts in this space.
I think the multi-scale perspective makes a lot of sense to me, and I think should not be abandoned. There's no single, scale that we should all be working on that matters. Some neuroscientists, scoff at MRI and sort of, whole brain human cognitive neuroscience for the reason that it's not neurons and not spikes. And that's a little bit misguided, because, it is what we can study with human beings that are, living and still behaving around us and so forth. So I wouldn't want to reduce everything down to neurons or spikes in the end. That doesn't make sense to me. So multiple scales should be maintained and are relevant for different types of questions we might ask. No doubt about that. Um, and then so you were asking about the
Well, how do we go about tangling with where that takes us, right? Because one of the things that we have been thinking a lot about is the different kinds of way that the system looks at these different scales.
yeah.
So if you just took spiking data from, let's say, like a little mouse that you have you have neuropixels sitting in its visual cortex while it sees a natural scene, so lots of complexity everywhere. And then you took something like a local field potential that you measured
Yeah.
from the cortical surface. These two signals look very different from one another. One of them's got these ballistic little punctate spikes. The other one's these kind of flowing, I guess potentials that are just rolling
Yeah.
along. And you make different inferences at different scales, maybe about the redundancy at one level or the kind of efficiency at another. And do you have thoughts about how to kind of grapple with this fact that the kinds of conclusions you'd make at different scales are different to one another? That it feels like there's a tension there, but maybe it's an illusory tension. I'm just curious your
No,
thoughts.
I think there's a tension, and in empirical work, we will not be resolving that tension in the foreseeable future because people have different take different methodological approaches, have different questions that research questions they might ask, and they necessarily aim at different levels of organization, I think. So that will always be with us. Again, I take refuge to computational modeling because that allows us to actually understand better how we navigate the different levels. We can, in principle, build a spiking neuron model, as some people have tried to do, as you know, of a whole brain, make it very realistic, and then see what kind of emergent signals we get from that, local field potentials, EEG type signatures, or even MRI or neurovascular coupling and MRI-like responses in a sort of synthetic framework, right? We build a system that is microscopically implemented, but then has macroscopic qualities and phenomenology. This can sharpen our understanding about how these levels interact with each other, I think, in a very intuitive way. So while I'm not advocating reducing everything down to neurons and synapses, having some grounding underneath those higher level signals is actually very valuable because ultimately that is what is happening, right? And having something underneath those higher level dynamic signatures, I think, is meaningful. But another thing about dynamics is that I have some very good colleagues who have take a very strong view about dynamics as being the only thing that matters. Okay, in the end, we don't even care where it's coming from, but it is sort of the dynamics that matters. It's the flow, it's the bifurcation, it's the this and that. Okay, and I'm not quite sure I would go that far. I certainly appreciate the dynamics plays an important role in brain function, and that can actually supervene and transcend, if you wish, connectivity constraints. It can jump over missing connections very easily and sort of link things up in a much more macroscopic way than connections ever could. But the connectivity ultimately is always going to be there, and without it, you don't get. I don't think it's very easy to get dynamics that is actually adaptively structured itself. You know what I mean? Like if you have no connectivity, or if your brain is randomly connected, you are not going to get dynamics to magically emerge that has any kind of adaptive validity to it, or actually is useful for behavior or for cognition. You still need the connectivity, even though there isn't a one-to-one mapping between the two, if that makes sense. And
I think that's spot on.
so I would resist playing one against the other. Is what I'm sort of
Mm.
looking at here. I've seen signs of that a little bit in our field where people take strong views on either of the two sides, and I feel like we don't have to do that. They are both aspects of brain function, brain structure and function, and they obviously collaborate
Mm.
with each other in ways that we are still not completely understanding. So I think both of them and their interaction in particular are important for understanding the brain.
So interestingly, a lot of these ideas that you're talking about now are probably most testable in the field of robotics. So I was curious whether you were kind of, as you've kind of gone through this really lovely, as you said before, non-linear circuitous path in your career, whether you're attracted to the idea of coming back to that space. I mean,
Oh,
I don't know if you've seen, there's some beautiful stuff in Japan at the moment
yeah.
where Juntani's doing this like
Yeah,
compositionality in
I know
robots, and
June.
it's
I visited
beautiful
his
work.
lab
Mm,
in Okinawa. No, I mean I'm still keeping one. I'm just peaking. I'm not. There's no chance that I will ever go back into this field. I'm too old now. But I'm still getting the occasional email and look at what's happening in that field, and it's fascinating. I agree with you. It's actually uh, for one thing, again, technology plays a big role.
Yeah,
It's
yeah.
a lot easier now to work in that area than it was back when I was PhD student and built a ridiculous looking tin can robot with wheels and a camera on top. So, that's where we're way past that now, and we can actually do things that are much more sophisticated, much more lifelike too, using biomaterials, using flexible,
mm,
compliant joints, things like that. This was unheard of 30 years ago, but now we can do it. This is fascinating to me. Um, I think that physical robotics, like building real physical robots, has still it's still hard to scale it up in terms of speed and agility and power consumption and so forth. So again, there's things we can do with simulation, right? And an accident that Randy B is a colleague of mine here, because his perspective was very closely aligned with that of many people here at my university and myself. I was actually on the committee that hired him,
oh,
which was a great success for us, I think. And so like what Randy does, right? He builds some things also now with 3D printing and so forth, but he still works largely with simulation because it allows him to have complete control and
mm,
perform controlled perturbations, experiments, and recordings. And we can do a lot. But again, let's not forget that the brain is just one part of the equation, right? The brain is connected to the body, is part of the body, never leaves the body in real life. It's always attached, always with us. We carry it around with us, and it feeds us our next input, right? One of the lessons I got from robotics was that, but just by moving our eyes in our environment, we are constantly changing the type of visual signal we're getting, and and therefore, of course, the functional connectivity resulting from that. So our brain, our bodies our bodies are sort of a mechanism by which our brain feeds itself data, right? And and that we can simulate. And I think, again, simulation and boiling complex issues down to simple implementations we can actually fully study in an embodied setting, but in a computer is actually very useful. And that is something I could imagine still doing, although, let's see. Um, um,
yeah, I
well,
was going to ask you the proverbial time machine question, Olaf. If you could start again today, where would you go? What would you train in? What would you sort of immerse yourself in? Would you like to go back to an area where there's no attention and focus and you can sit and marinate and wallow in the kind of exciting ideas in your own time? Or
yeah,
would you like to do more of this kind of connector hub between a centrality maximization where you're
that's my
thinking,
style.
yeah, yeah.
That's
Okay.
really my style. I'm not a specialist by inclination. Don't see myself spending 30 years on the same thing. I do enjoy branching out and um, learning new things in the process. And so that was sort of the path I've taken, perhaps with a somewhat disregard for my professional future. Again, I got my first job fairly late. I had a 10-year postdoc, as I call it, which was basically true. I didn't have a real job until 10 years out of my PhD, in part because I was so engrossed with all these different things that I was doing with neurons, with robots, with information theory and whatnot. And it was hard for me to settle down on one thing, because when you do get a real job, right, they want you to have like this is your grant, right? This is what you're going to write in your grant. And I had a hard time getting to that point where I had enough of a solid focus to really be able to pull that off. So that's the downside. But no, I really wouldn't do anything differently. I was extremely lucky in many ways, by having made some choices that turned, having put my money on things that in the end worked out okay. Um, but um, maybe that's partly luck, and maybe it's also partly because you work on it. That's why they work out, right? You kind of pour yourself into it, and you keep hammering away at it, even though nobody wants to listen. You keep hammering away at it. And it took us 8 years to get the first brain network paper published. We had it basically written in '92, and then it took us 8 years to get it out in 2000. And instead of giving up, we kind of persisted on it, and that paid off. So, um, I don't know what the next big thing will be. That's the other thing. I mean, networks are now so baked in to uh, to our field, and I was on a Zoom call the other day, and someone mentioned the connectome and asked me whether I had heard of it. It was funny. I mean, it's 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 so baked in now. Everybody seems to know what it is, and that was just not the case 20 years ago. It's
Like, yeah, I've heard of it.
yeah, it's amazing to see that happen, and I never would have believed it would take off the way it did, but it did.
That's
Yeah.
great.
How long was the debate and the thinking time before, deciding on the word to call it? Because, it's a big thing when you name something, and,
Yeah.
with it, how long
Yeah.
do you think tossing it around?
Well, connectivity has been something we've been working on for over 10 years, but we wanted to make a complete map. And I remember it was really me who came up with the connect home as a word. I shopped it around to some people who shall remain unnamed, and they all turned me down. They were saying like, "Oh, you're crazy. This is a joke or something." But I felt it was worth labeling it that because, like the genome, it is a foundational data set for biology. It is structural in nature, even though the structure is much more complicated than just A, T, C's, and G's.
Yeah.
And it's a foundation for neuroscience. That was my idea back then. But it took us 2 or 3 years of writing various versions of the white paper and then rotating it among colleagues, getting input, eventually finding a place to publish it.
Yeah.
And then again, after that, 3 or 4 years, stunned silence. Nobody took it up for quite a long time, and so but then it did take off. Yeah. Julia.
Yeah, I have a question on behavior. So it seemed that, right, you had your intuitions about neural networks from observations on how your architecture changed, the visual perception grouping that you showed us. So it started from behavior. You saw something in the behavior, and then you had intuitions on how, the architecture of the brain may be working. And I wonder nowadays, right, we don't really do that anymore. It's like kind of a niche. And I'm talking about network neuroscience. Most people don't really start from behavior, or they don't do cogneuro. It's mostly, I would say, relegated to resting state or task based, but they don't really have that intuition. And maybe it's biased by my own training. But I was wondering if that should still remain our ground truth. Because, when we start abstracting and doing stats and blah, 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 we kind of lose track of what are we contrasting our theory against, and what should be the end goal. So I wonder if going forward, back to what Mayuri was asking you, is sure, we may have better technology, better, tools to look at data, but maybe what we should be doing is trying to understand the behavior, the outcome better, and contrast these, very high level theories of the brain to better concepts of behavior as well. And yeah, I was just wondering where you stand on this.
Yeah, I from my vantage point, it looks to me as if resting state is still very important, gives us a lot of information about how the brain is sort of organized, sort of a ground state, and it's quite dynamic in its own right. But I think there's a lot of movement now to go to more ecologically valid paradigms, right? Movie watching, some forms of spontaneous behavior can be carried out in the scanner. In some ways, the scanning environment is not conducive to it. It's prevents us from engaging in natural behavior in some ways. So, but I think there is a lot of movement in the field to go a little bit away from resting state and approach sensory perception and behavior once again in a much more rich setting. So that's good, I think. But the technology, especially with MRI, will be cumbersome for quite some time. So maybe other modalities will become more important again, like high density EEG or MEG. I don't know. But we are limited to some extent by our technological, but the methods that we have that somehow get in the way of observing natural ongoing behavior. That I think in the end is the holy grail. I think, we do want to know how the brain operates, how it changes, how it interacts with the body to give rise to rich behavior and rich behavioral output. That that's a goal. It's a good goal to have. I think we're getting closer to it. Resting state was important. It has given us a lot of really important insights. I get that about the limitations of it. I sometimes jokingly say resting state is really a misnomer. The brain isn't really resting, and it's not a state. It's not something static. It's actually very dynamic, even under those very impoverished conditions. And I think that's opened our eyes and sharpened our tool set for understanding dynamics, for measuring it, for tracking it. These are really important tools to have when we go to actual behavior at some point, hopefully.
Isn't that the funniest thing, Olaf, of our field? I think in part because neuroscience is still so young, we're really, really bad at naming things. And so functional connectivity is just one of the most awful terms ever.
Oh, I blame Carl all the time.
But we're pointing at something interesting, right? We're pointing, here's a
Yeah,
signal, here's an important feature, and that's true, but you
yeah,
can't take it too literally. And I think we need to be kind to ourselves when we come back and we have this kind of revisionist history and we say, well, actually, that's not really the right way to think about it. We thought it was this
yeah,
one thing. We thought it was a baseline resting state, but no, it's not. It's now much more dynamic and it's much more about
yeah.
internal constraints rather than external ones. And that's good. And let's use that information going forward. So
And
I think
it's at its base, it's a very simple statistical construct. I mean, it's just
yeah,
statistical dependence. So it's not even, there's no causality implied, nothing. So there is a lot that is lacking. And yet, it is what it is. So I'm not advocating we should abandon it or anything. It's
mm,
just we have to, I think again, we have to educate ourselves in our scientific training, in our practice, to always remind ourselves where do these constructs come from,
hmm
one of my pet peeves, the things like I do in my talks now is I show them dynamic functional connectivity during resting state, and I ask the audience to track, the default mode network, like where is it? Okay, let's track it. And the point of this little exercise is that it's not always there, it sometimes just disappears completely. So when we talk of it, we give it a name and we say that's what it is, and we put lines around it, and that is where it is in the brain, we have to remind ourselves that it is, on average, it is there in that location, but it's not always in that configuration. It's not always even present. So it aggregates, it dissolves, it recombines with other parts of the brain, etc. So it's much more dynamic, and we are losing the dynamics when we are giving things names and we point to them as if they
yeah.
have physical presence, okay, which in the case of a functional connectivity network, it doesn't. It's statistics, it's correlations.
It's one of the reasons that I love and really advocate for multiple methodologies when we're analyzing
Yeah,
data, because you can't help but realize that it's
yeah,
completely depends on the method you use, the type of
absolutely.
answer you get. Yeah, yeah. So did any of the students that haven't had a chance to ask a question, Ruby, is there anything you wanted to ask, or Julia, Murray, Claire, not a student, but anyone have any final questions for Olaf? I'm wary of the time. It must be getting kind of late. I should ask Logan and Brandon with their cameras off if you guys have questions as well. Please feel free. to ask some last ones. If not, we can sort of start to wrap things up. Anyone?
Anything you wanted to say,
Yeah?
Olaf? Anything that you were kind of, because I feel like when we asked you to speak at this, it probably caused you to kind of think a little bit big picture and reminisce. Was there anything that you kind of thought of when you were kind of mulling this idea around in your mind that hasn't come up that you want to say?
I don't know. I mean, no, I mean, I think we've talked about a lot of things. I'm reminded of how different the whole landscape looked like when I was in your shoes as students and postdocs. I think I was much less worried about my future in terms of career path and positions and so forth than I now see my trainees. They are very worried, and I think understandably so, about what their next step is. And it's very there's a lot of pressure on them now, to become proficient in a certain part of science and to publish continuously and without interruption and to have ultimately have grants and all of that. And I never felt that pressure as much. It was more internally. I wanted to publish. I wanted to write something, but not because somebody told me I had to. I wanted to because I had something to say. I felt at the time, and five people listened, so that was great. So, you know, and so that worries me a little bit because I think it takes away from the on what science perhaps should be as an intellectual enterprise, as something that is above and beyond just a job. It's as scientists, we have particular responsibilities as well. We are, pursuing knowledge, knowledge hopefully that will last and that will make a difference in the world. We are in a long tradition of hundreds of years, of thousands of years of scientists who have come before us. And if we force our trainees to think too much about, the job and the paper and the grant and whatever, I think we are taking away that fascination and that important. Scientists are different from other people, in some ways, and they should be.
Mm.
It's not just a job; it is more than that. And I came up in a landscape where that was more like that's what people around me thought, I think. At least at the time, I was surrounded by people who had similar vision. And it made it easier for me not to focus on the next paper and the next hot topic and nature and science and whatnot, which again, I never had. But focus on the problems and on really, really trying to make a contribution. It may end up being nothing in the end. Who knows? That's the risk, right? We don't know. In 50 years, somebody else comes along, and all we've been talking about today might just turn out to be complete bogus. It's possible.
Yes, what an honor, Olaf, to have helped them come to the actual right answer.
That's the way it is. That's science for you. I mean, there's lots of people go down the wrong path. That's the risk you take, but at least you do it, and you hold your head on high and you do it, and that's I think what we should do as scientists. So I'm saddened sometimes to see students be so disheartened already at a very young age, and become cynical about it and think of it as just a deal, or something that they have to be doing. If you just turn out the papers and get the grants, then everything will be fine. That's not really what science is about, It's about making a contribution and advancing knowledge, and I think that gets forgotten, unfortunately.
Well, I couldn't agree more, and
yeah.
I think you've over the last hour and a half, you've demonstrated so many of the things that I admire about you as a scientist, Olaf. Your curiosity, your willingness to pivot and change when the signal changes, and your willingness to communicate what I think are really deep, important messages for us to hear, not just as a group of scientists, but probably as a world right now, that we really need to think carefully about what we're aiming for. Otherwise, we're going to crash into a brick wall. So
tell me about it. Yes.
So
Thanks
with
for
that,
reminding me.
yeah, well, back to reality, right? No, look, that was just fantastic, Olaf, and thank you so much for taking the time. Hopefully we'll get a chance to chat soon about science, and I'll keep reading the beautiful stuff coming out from the talented young people in your group. So thank you so much.
For
Thank
sure.
you.
Thanks so much, and have a good rest of your day.
Thank you.
Bye-bye.
Thank
See you, mate.
you.